All right, well, we are at 8 a.m., so why don't we get started? And delighted to have Dr. Steve Barzy, our interim division chief for geriatrics and gerontology, introduce Dr. Tobey Betthauser, another faculty member in geriatrics and gerontology. So, Steve?
Thanks, Lynn. And welcome everyone to Department of Medicine Grand Rounds for today. I'm excited really to be able to talk a little bit about our my wonderful colleague and neuroscientist who has been within our group now for some time. So Dr. Tobey Betthauser is a medical physicist. He is a neuroscientist, and as I learned earlier today, previously an audio engineer in a different phase of his life. And he today will be talking about, I think, some really pivotal work he's done. But to start, he has a background in medical physics, where he had done his undergraduate training in SUNY Plattsburgh, and then joined us here at the university, where he has served in various capacities, but has worked with Dr. Sterling Johnson, whom most all of you are well aware, and really has done some pivotal work, pioneering work, I'll say, in the area of biomarker modeling. Not only is he a wonderful scientist, however, though he is also a really amazing colleague who collaborates with many of those, both within the Alzheimer's Disease Research Center, but also really across campus and across the country. And he's going to be sharing a little bit about, we've all changed our frame of reference with regards to how we think about dementia, now much more so as a chronic illness that has decades of lead-in. And for us to understand what that trajectory is for the illness early or upstream, and how we can then influence risk factors and better understand the illness, is going to be really pivotal as we move the needle on managing persons living with memory illness. And so without further ado, Tobey, thank you for being here today. And please share on biomarker clocks in Alzheimer's, predicting dementia risk and timing.
Thanks, Steve, and thanks for the invitation to present today. So I wanted to just show conflicts of interest. I will be discussing some drugs, in particular PET tracers, that are not yet FDA approved. And I'll be discussing some topics that are kind of at the razor's edge of research. And so these are some things that probably not quite into the clinical standard of care. For learning objectives today, we kind of have our standard objective of understanding some current updates and how to apply these things in clinical practice and research. But more specifically, I want to go over how we can interpret amyloid and tau PET imaging, as well as plasma phospho-tau 217 in a longitudinal and dementia progression context. And then also give you some information about kind of the typical progression of these biomarkers, how these things can be a little heterogeneous between individuals, as well as how these things can differ between some populations. So before I dive into all of that, I just want to briefly acknowledge the many people that contribute to these studies. Anybody that's been in this field for more than about a month knows that this takes a massive amount of resources. Not just funding, but staff and a number of investigators. And we're very fortunate at Wisconsin to have a very collaborative and large investigator team and a lot of staff and other leaders that really drive this research. And of course, importantly, perhaps most importantly, the research participants. And the work I'm presenting today is several thousand participants across multiple studies. And additionally, I just also want to disclose that I received research funding from NIH, and I've received travel support from the Alzheimer's Association, NIH, and I'm a consultant for the Harvard Aging Brain Study. I also wanted to give just a brief background of my connection to dementia. So this photo was taken in 2010. On the left is my late grandfather, Grandpa Lyle, and my grandma June, both of whom developed clinical impairment over the course of their lifetime. My grandma's was probably more vascular dementia. My grandfather was diagnosed with Alzheimer's disease in 2013 and died about 9 years later. So when I see this photograph as a dementia researcher now, the thing that I wonder is, when did this disease start for him? And knowing what I know now, it's quite likely that, unbeknownst to us at the time that this photo was taken, it's quite likely that he had proteins developing in his brain of Alzheimer's disease at that time. And this really drives a lot of my motivation to understand the timing of pathological events in Alzheimer's disease and how these influence symptoms that we see. And then also just kind of a brief overview of Alzheimer's disease and where we're at today. So these are facts and figures that the Alzheimer's Association publishes annually. And it'll be no surprise to anybody in this audience that Alzheimer's disease and dementia is a growing public health concern. These are kind of where we're at today in 2020, with about 6.1 million people living with Alzheimer's disease. And this is projected to increase over the coming decades and become, especially amongst those in the older demographic in our population. Along with this increased number of people with dementia, there's a substantial financial drain to care for people with Alzheimer's and dementia, estimated to be $384 billion in 2025. Importantly, about a quarter of these costs come out of pocket. And this also doesn't include the estimated almost 12 million unpaid caregivers. Again, anybody that's had any relationship with people with dementia know that this doesn't just affect that person, but really their entire care support team, including family, friends, and others. So, and specifically in Wisconsin, it's estimated that there are about 205,000 unpaid caregivers caring for people with Alzheimer's and dementia. When compared to some other causes of death, one of the things we've seen over the past 20 years is that the death due to Alzheimer's disease and associated causes has steadily increased over this time span, whereas other leading causes of death have remained relatively flat or have seen marginal increases or even decreases in some cases. When we focus on the disease pathology, the disease neuropathologically is defined by aggregated beta amyloid plaques, which are in the extracellular space, and neurofibrillary tangles composed of tau protein, aggregated tau protein, and these are within the neurons. And again, just kind of going over some of the basics, we know that increasing number of tangles and neuritic plaques in isocortex are associated with more advanced cognitive decline. This is a figure from Peter Nelson's paper from 2012. And then we also see increased levels of atrophy, brain atrophy in those with AD pathology. I also just want to briefly go over tau pathology because it's relevant to the biomarkers we're going to discuss. The idea is that the tau protein has a normal role in stabilizing microtubules, but in disease becomes hyperphosphorylated, which leads to microtubule destabilization. This ultimately forms these soluble protofibrils, and then later this leads to aggregation of paired helical filaments, which are insoluble, and then later goes on to form these neurofibrillary tangles. This is important as we get into understanding some of the current biomarkers. And when we think about tau spatial progression, we see this kind of stereotypical pattern wherein tau aggregates start really in the medial temporal lobe, particularly in entorhinal cortex and hippocampus, and then spread kind of outwardly into the temporal cortex and larger neocortex in advanced stages. So shifting to biomarkers, we're fortunate in the past 20 or so years that there's been great developments in biomarkers. So we can measure amyloid and tau pathology in vivo using PET imaging. So on the top showing examples of amyloid PET scans in an amyloid negative and an amyloid positive case. Bottom showing tau images for a tau negative and a tau positive case. We've also been able to measure these in cerebral spinal fluid, and more recently there's been a lot of development in the blood biomarker space. And I think the thing to keep important in your mind here is that these are really kind of macroscopic measures of really like microscopic detail, right? So there's always going to be some detection sensitivities and other things involved in these, and it's there's also some pathological inference that we can make, but sometimes we don't always know what's driving these biomarker signals. When we think of the typical biomarker progression in Alzheimer's disease, this was made famous by Cliff Jack. So the idea is that we can detect levels of abnormal amyloid first in typical disease, followed by tau positivity, neurodegenerative changes, and then later we see clinical and cognitive impairment. And we largely break this disease into kind of these two phases, the preclinical phase, meaning before clinical symptoms. And this is the phase where we can measure abnormal levels of these biomarkers, reflecting amyloid and tau pathology. But we don't yet have meet the bar for clinical impairment. Versus the clinical phase being, we can now detect clinical symptoms within patients. And these research over the past 20 years has led to kind of these evolving frameworks of how we think about diagnosing Alzheimer's disease. So this was just updated again last year, which is part of an Alzheimer's Association working group with input from the NIA. And we largely now kind of focus on this rubric for defining AD in terms of a clinical stage and a biological stage. So just to kind of go over this rubric and how it's applied across kind of the horizontal axis here, we have clinical stages. Stage 0, I'll describe in a moment, but clinical stages 1 and 2 are essentially in the preclinical phase where stage 2 is kind of a transitional phase where people are maybe starting to notice some subjective cognitive complaints and mild cognitive symptoms, but these don't yet meet the level for clinical impairment. Whereas clinical stages 3 through 6 are kind of advancing into clinically detectable levels of impairment and progressive dementia. And then on the vertical axis is defining biological stages based on amyloid and tau pathology. So the idea is that stage A and the diagnosis of Alzheimer's disease can be made by a positive amyloid biomarker. I could give a whole hour-long lecture on just this framework, but this is kind of a little bit of a simplification. But the idea is that people will be amyloid positive, but tau negative in this early preclinical phase, and that tau will continue to spread, again, beginning in the medial temporal lobe and then out into the greater neocortex. And these represent the more advanced biological stages. And the way that this rubric is applied is that anybody that goes along this diagonal is thought to follow kind of the stereotypical progression of Alzheimer's disease. So we expect kind of these biological core stages, the biological stages on the left to correspond to the clinical stages on the top. But it also allows for people to have more advanced clinical stage for their given biomarker stage. And the opposite, people can have a less advanced clinical stage given their biological stage. So it allows us to kind of characterize people that have more advanced progression given their biology or their biological stage, which is a key thing that we're trying to understand as a field as to why one person goes through an accelerated path, whereas maybe other people are more resilient to pathology. In terms of drug approvals, it's been a very exciting time to be in the field. The past couple of years, we've seen 2 different drugs approved for anti-amyloid therapy. And kind of the notable difference in these drugs versus what's been previously approved is that these seem to actually slow the progression of disease rather than just treating symptoms. The other pattern kind of in clinical trials over the past 10 to 15 years is kind of moving earlier and earlier in disease to treat and when we can treat. So we still do not have any approved drugs for the preclinical phase of AD, although there are ongoing clinical trials. And just to kind of summarize the results from the donanemab and lecanemab trials, so lecanemab on the left, donanemab on the right. So each of these is showing amyloid burden measured in a unit called centiloid. And a centiloid essentially is a standardized unit across all amyloid tracers where 0 effectively represents the average of a young control group thought to be amyloid negative. And 100 is kind of the typical level of amyloid that a dementia group would have. Most research will say a positivity threshold around 20 to 25 centiloids is reasonable. And what you can see from these trials is that both of these monoclonal antibodies targeting beta amyloid effectively remove plaques over the scope of the trial. And both of these drugs met their primary endpoints in terms of a slowing of clinical progression based on the clinical dementia grading sum of boxes. Another factor that came out of the donanemab trial was that it appeared that people that had low to medium tau had a slightly greater treatment effect compared to when they combined the entire population in the treatment arm. So kind of some indication that, again, kind of treating earlier in disease is likely beneficial. And then this past summer, this was updated. I couldn't find a nicer graphic, unfortunately, but in a study from Eisai, they presented at the Alzheimer's Association International meeting where they took participants from the Alzheimer's Disease Neuroimaging Initiative, as well as the BioFinder study, and matched these two participants that were in the lecanemab trial. And the idea was they wanted to see if people that were on the lecanemab treatment arm continued to show clinical benefit extending well beyond the trial. The caveat with these results is, we don't have an actual control group anymore in this study, so we're kind of extrapolating from observational cohort studies what we think those groups would have done. But still somewhat promising that we think that these drugs may have a lasting effect even after the initial treatment window. So this kind of brings us to current day, where we're at, which is we really need to understand when in disease to treat, especially with these anti-amyloid therapies, to maximize the effectiveness of these treatments. I would say the field in general is moving towards a more personalized approach to medicine, which really shouldn't be surprising to anybody, particularly if you look at oncology, where the better tests that we have, the more predictive we can figure out how those tests can tell us about disease progression, the better we can personalize our treatment approaches and prognosis. And in order to do this, I would argue that we really need to understand the things that accelerate and slow these biological and clinical progressions. So my lab, I started in the middle of COVID, about a week from when Lynn started as chair of the department. It was an interesting time to start a lab, but I'm very excited about the work that we're doing. The overall goal of our lab is really to develop accurate, generalizable, and clinically relevant tools that we can share in an open source way that will really inform AD prognosis and allow us to take these biomarker results, see what replicates across multiple different cohorts, and develop risk models that are tailored to different population groups. In addition, we're really fortunate to have the resources and study cohorts that we can also leverage some postmortem validation studies where we can look at people that have had either PET imaging or fluid biomarkers before they died and come to our brain donor program. And this really helps us anchor and understand why we see particular longitudinal patterns in our biomarker data. So getting more specific into this idea of how we study time and contextualizing this, it's kind of been established for over a decade that this preclinical phase of Alzheimer's disease is thought to last 20 or more years. And to put this into context with some of the developments here, I'm showing based on a timeline of FDA approvals. And then on the top, showing kind of when certain discoveries happen. So for example, amyloid PET imaging has only been around in humans since about 2004. So again, if we think about the fact that this disease has this extended 20-year period of a preclinical phase, this really poses a challenge for studying Alzheimer's disease, because even had we started massive cohorts the day that amyloid PET became available with PIB, we still would potentially just be catching people converting from the preclinical to clinical phase. So, to further contextualize this, if we look at the discovery of tau PET in humans, this has really only been around for a little over 10 years. So again, if we're trying to understand this full 20+ year disease timeline, we haven't really even had the tools to do it, to be quite honest. And I will mention, if we look at from when these things are discovered to FDA approvals, we also see kind of, this offset here with amyloid PET, with tau PET, and then more recently, just this year with some of the first blood tests being FDA approved. Now, contextualizing this further, if we look at our observational cohort studies, what we see is that we don't typically have even more than about 10 to 15 years of longitudinal data, right? So this is kind of what facilitates the needs for these methods to understand this prolonged period of disease. And really the idea is that we want to take these longitudinal observations and turn this into some biomarker-based timeline. And so that kind of brings up this idea of what is a biomarker clock? And kind of an oversimplified version is it allows us to take these age-based longitudinal measures and recast disease into a biomarker-specific timeline based on how long that biomarker has been abnormal. And I would say the bulk of this work in our field has been done in amyloid PET imaging. There's been several methods proposed to do this over the past decade or so. Largely, these models show very consistent things, which is that you can use multiple different methods to get at this idea that there's kind of a normal pattern of accumulation once positivity occurs. And the method that I developed is called sampled iterative local approximation. And I just go through kind of a toy example here of how this works. So on the left, we have our observed amyloid versus age data. This is data from the Wisconsin Registry for Alzheimer's Prevention. We are able to apply the SILA algorithm, which outputs a curve which tells us how amyloid changes over time. And then we set the 0 point of this to be at some level in which is indicative of an abnormal level. So we call this amyloid positivity or positivity threshold. And then once we have this model trained, we can apply this to individual level data. So in this case, I'm showing this is an individual scan from a single participant. So here shown on the age-based timeline, here shown on the amyloid-based timeline. And we're able to take that value of amyloid at that time, project that down to figure out how long they've been amyloid positive. And then by subtracting that from their age at that amyloid scan, we can estimate when amyloid positivity occurred in this individual. So the really kind of neat thing about these methods is that while the models themselves are trained on longitudinal data, we really only need one observation to be able to estimate when people became biomarker positive. And in the first study that we had on this, we had several aims. One was just to compare multiple different methods of doing this, and largely saw that this was consistent. We tested this in 3 different cohorts. So again, ADNI, the Alzheimer's Disease Neuroimaging Initiative, the Baltimore Longitudinal Study on Aging, which is BLSA, and then the Wisconsin Registry for Alzheimer's Prevention. And amongst the things that we did to validate these methods, we were able to show that we can estimate antecedent amyloid levels with high accuracy. In people that were observed to convert from negative to positive, we were able to show that we can estimate that onset age, usually within about a year or two. And importantly for these models, in all 3 of these cohorts, we didn't see an effect of ApoE or sex in terms of how fast people accumulate amyloid. And in ADNI, where we had enough sample to look at dementia, this also was not associated with differences in amyloid accumulation trajectories. Applying these methods, then we started to look at factors that associate with when amyloid positivity occurs. Not surprisingly, we found an effect of ApoE genotype. So these are shown on the left here for individuals that became amyloid positive. These are the ages that they became amyloid positive on the left, organized by ApoE genotype, kind of going from least risk with E2, E3, and E2 carriers to E3 homozygotes and into E4 carriers and homozygotes. So we see this expected pattern where people with increasing number of ApoE4 alleles have an earlier amyloid onset age. But something that jumped out in the results as we were looking at this was the wide variability in when people become amyloid positive, even within, say, ApoE genotypes. So here we're seeing roughly a 40-year span in which people that are E4 homozygotes become amyloid positive. Another thing that these methods allow us to do is to study the time from amyloid positivity to dementia and things that speed up or slow down that timeline. So really getting at things that accelerate or slow the preclinical time frame. So in this study, this is ADNI data, again, where we had enough participants convert to dementia during the course of study. We saw that APOE4 carriage and older age at amyloid onset, and females had a earlier transition from amyloid positivity to dementia. And notably, when we kind of compare these effects across these different variables, we can see in particular people that became amyloid positive kind of in their 70s and early 80s had about a 4-year shorter time frame from amyloid positivity to dementia, which contrast with the ApoE-based and sex-based risks was about twice as much in terms of how much it accelerated progression. The other thing that jumped out to us was that E4 carriers kind of have this double hit. So not only do they have a faster preclinical time frame, but they also get amyloid earlier in life. So if we're thinking about lifetime dementia risk, right, the earlier that you develop pathology, the more likely it is that you'll develop dementia. But then also by being an E4 carrier, you have both of these things combined to really shorten that time window and increase your lifetime dementia risk. And these are results that have largely replicated across multiple cohorts. So I'm just throwing here 3 examples from 3 different studies. And again, another theme that pops out is the amount of variability in the time from amyloid positivity to dementia. So for example, in ADNI here, we have some people that are experiencing dementia essentially at the same time that they're getting amyloid positivity. These are maybe not due to Alzheimer's disease, but then we also have people on the other side of the spectrum who can make it 15, 20 or more years with amyloid without developing dementia. And so these sources of variability are things that we're really trying to understand in our current research. So I've shown you that applications with amyloid PET, but there's several other biomarkers that we could potentially develop clocks for that would allow us to study disease. And so this is just kind of a brief overview of the things that we've attempted and what has worked. And I'll focus kind of the rest of my talk on applications with tau PET imaging as well as plasma phospho-tau 217. So if we look at the tau PET-based models, so similar to amyloid, we're not seeing effects of age, ApoE, or sex in terms of influencing these overall trajectories. But we do see some regional differences in performance. So for example, if we look at kind of how well people align to these 2 modeled curves, as well as in the statistics over here, that if we're using kind of a larger region of interest that includes medial temporal and neocortical brain regions, the model performance is a bit better compared to when we just use, for example, entorhinal cortex and trying to get at kind of these earliest changes in disease. When we look at people that were observed to convert from tau negative to positivity, we're able to see that these estimates are usually within 1 to 2 years of when the window in which they converted. The caveat being we don't know the actual age they convert just because of variability in data and sampling frequencies and these types of things. And then something that was unique to tau imaging that we didn't observe with amyloid data is this effect of dementia, particularly in the entorhinal cortex. So here, each of these lines is showing a modeled curve from tenfold cross-validation, and we see kind of this divergence in entorhinal cortex, but not really metatemporal ROIs. When we get rid of or when we exclude people with dementia from the analysis, we see much better concordance amongst those in the entorhinal cortex. So in looking into this a little bit further to try to understand what's going on here, we're also seeing basically that people with dementia are much more likely to have negative changes in their tau burden in the entorhinal cortex. And so this is likely making it challenging to estimate time estimates in entorhinal cortex for people with dementia. And one of the things, kind of getting back to this idea that how do we understand these longitudinal changes in a pathological context, we have a study ongoing where we're taking individuals that came to brain donation that had antemortem tau PET imaging to understand the pathological correlates of these biomarkers. And our current hypothesis is that um, essentially this decrease that we're seeing in tau PET signal might actually be due to the fact that the tracer doesn't bind as well to ghost angles, which occur after the neurons die. But this is ongoing work that we're continuing to try to figure this out. In another study that was led by Margot Heston, who was a postdoc in my lab, we'd applied these tau clocks to understand when tau positivity occurs, what factors are associated with that, and if there are any factors that speed up the time from tau positivity to dementia, and also contrast some of these different timelines, so age, amyloid, and tau-based timelines. So again, this is using data from ADNI. So if we look at kind of the overall population, the median tau negative survival, or I guess the age at which half of the population became tau positive, was 88 years, which is probably a good thing just based on overall dementia risk. And then we looked at factors that were associated with tau onset age. We saw again an E4 effect. So individuals with higher numbers of E4 alleles had an earlier tau onset age. Individuals that had higher levels of amyloid at tau positivity also had a slightly earlier tau onset age. Females compared to males had a slightly earlier tau positive onset age. And then we also see kind of this, well, hypothesized as a selective vulnerability and also kind of a survivor bias, in which people that were in the 75 to 85 year age bracket have this higher level of risk of developing tau kind of within the next 5 to 10 years after their baseline tau PET scan. In addition, we were also interested in trying to understand if select social determinants of health impacted when tau positivity occurs. And so we looked at 2 different measures, the American National Adult Reading Test, shown on the left, where higher total errors are associated with lower exposure to literacy enrichment. And then on the right, just based on educational attainment. And the results are rather consistent, but somewhat modest effect sizes, which is higher total errors on INARD, as well as lower education were associated with earlier tau onset age. So this is an active area of research where we're trying to understand some potential connections between the social determinants of health and biological mechanisms that might influence biomarkers and when they become abnormal. And then if we again start to look at the variability, so similar to amyloid, we're seeing high variability in tau onset age. Again, ApoE was kind of the strongest predictor amongst all of these things. And we still see kind of that 30 to 40 year window in which people develop tau positivity, even within, for example, the E4 homozygote genotype. Now shifting to looking at the time from tau positivity to dementia, here we see a much more truncated timeline. So half of the people that became tau positive in this study experienced dementia within just under 7 years of tau positivity. And when we looked at factors that accelerated this, we saw an effect of amyloid, such that people with higher levels of amyloid burden had a faster decline from tau positivity to dementia. And then also people that developed tau pathology at an older age also had a much shorter time from from tau positivity to dementia. And then kind of in the last aim of the study, we wanted to contrast these different time-based metrics in thinking about how this can inform dementia risk. So this first plot, again, is showing CDR sum of boxes, so higher levels indicating worse dementia, and a level about 4.5 kind of being the threshold for mild dementia. So here, just looking at the data, you can see there's kind of a wide range in which people cross this 4.5 line. It spans several decades. If we start to look at an amyloid-based timeline and then a tau-based timeline, we see kind of this progressive shrinking of this window in which people can develop dementia after amyloid positivity and after tau positivity. And if we restrict this to people that were observed to become positive on both amyloid and tau PET, we can kind of more directly compare the ranges of these. So for example, if we're looking at age of dementia onset in this group, we see this spans about 37 years, whereas on an amyloid-based timeline, this shrinks down to about 25 years. And in a tau-based timeline, it's shrinks again down to about 15 years. And now this is kind of really where we're trying to head with these models, is to get time-based predictions or probabilities for when people will develop dementia. Currently, these are anchored to these biomarker timelines. So on the left, again, this data from ADNI showing time from amyloid to dementia on the left, time from tau positivity to dementia on the right. And then the y-axes here are showing probabilities of remaining dementia-free. So 1 being everybody remains dementia-free, and 0 being everybody has dementia at that point. And if we contrast these two timelines, again, we see this expected pattern, but it's really quite dramatic. So for example, 5 years after amyloid positivity, about 90% of the study population remains dementia-free, whereas 5 years after tau positivity, less than well, about 30% of people experience dementia, so about 75% remain dementia-free. If we push that out to 15 years after amyloid positivity, we see about a fourth of the population has experienced dementia, whereas at 15 years of tau positivity, virtually nobody remains dementia-free. So this is kind of where we're headed in terms of the prognostic value of these methods, is to be able to put people on these timelines to give them a sense of what's the chance that you'll develop dementia in the next 3, 5, 10 years. If we kind of stratify these curves a little bit more, on the left now we've separated people out in terms of those that are amyloid positive but remain tau negative, and those that are amyloid positive and become tau positive. We can see a dramatic difference in the dementia-free survival. So for example, 20 years after amyloid, we're seeing almost 3/4 of the participants remaining dementia-free, and versus those that develop tau, it's less than half. So really kind of developing tau obviously is a big additional risk factor in terms of accelerating dementia risk. The other thing we can see in both the tau-based and amyloid-based timelines is that these colors indicate the age at which either amyloid positivity or tau positivity became abnormal. And you see again this pattern where people that are younger at amyloid or tau positivity essentially can make it longer to dementia after these events, but those that are older decline much faster. So again, this is something that we're continuing to see consistently across biomarkers, and I think something that will have high prognostic value as well. To contextualize these results, again, thinking about these kind of timelines. So this is now showing tau burden as a function of amyloid positive years. And again, we can see kind of in this first 5 to 10 years that there are not that many people that are tau positive, and we can see quite a bit of variability even before and after that in terms of when individual people start to become tau positive after amyloid positivity. Okay, and then so getting to the plasma, kind of shifting gears. This paper was recently accepted this week, actually, which shows that we can also apply these models to plasma phospho-tau217. So in this work led by Carly Cody, who's a former PhD student in our neuroscience program in Sterling's lab, has led some really awesome analyses showing that we can apply these models to 2 different assays that measure plasma phospho-tau217, and that perhaps with a little bit more variability, we can estimate the age of plasma phospho-tau217 positivity as well. And one of the neat things that this allows us to do is now to contextualize biomarkers with respect to each other, right? So here on the left, we're showing plasma phospho-tau217 levels as a function of how long people have been amyloid PET positive. And you can see for the most part, people remain phospho-tau217 negative prior to amyloid positivity. And for the most part, people become positive on these after amyloid positivity. And if we do the converse and we show amyloid PET burden as a function of plasma phospho-tau217 positivity, we again see that people becoming amyloid positive before plasma phosphatau-217, but also quite a bit of spread in terms of when these times happen. We can then use these timelines to look at other aspects such as tau PET positivity and entorhinal cortex and getting out into the neocortex in the inferior temporal lobe. These are data from RAP and our Wisconsin ADRC showing kind of this typical progression of amyloid PET first, followed by plasma phosphatau-217 positivity, entorhinal tau positivity, and then spreading out into the neocortex. If we recontextualize this based on how long people have been plasma phosphatau-217 positivity, we can see kind of this progressive pattern of plasma first, followed by entorhinal tau and inferior temporal tau. So again, the nice thing about these models is that rather than kind of having things in like a biomarker concentration or these kind of SUVR Centiloid units that require a bit of expertise to interpret, these time-based metrics really kind of give us a sense of how long it takes somebody to progress through disease on average. And similar to past studies, we're seeing that compared to an age-based timeline, amyloid and phosphatau-217-based timelines have a stronger association with preclinical cognitive decline. And in this case, we saw that depending on the assay, plasma phosphatau-217 time either had a similar or higher association with preclinical cognitive decline compared to amyloid PET. And then the last study I want to highlight, or last group of studies, is related to Down syndrome. So I mentioned way at the beginning this idea of stage 0. So stage 0 is really meant, clinical stage 0 is meant to represent people that are thought to be genetically predisposed to having disease, having Alzheimer's disease. And Down syndrome is characterized by a triplication of chromosome 21, which encodes the amyloid precursor protein gene. And effectively, this results in an overproduction of APP and results in individuals with Down syndrome getting amyloid earlier in their life and nearly 100% penetrance of dementia during their lifetime, with most individuals with Down syndrome developing dementia by age 55. So this is some ongoing work that I've recently started working with Dr. Brad Christian's lab and Dr. Matt Zammit in starting to contrast the time course of biomarkers in Alzheimer's disease in sporadic late AD in a neurotypical population to our Down syndrome cohorts. So in two recent studies, one published by Dr. McVeigh, or PhD candidate McVeigh, we're seeing actually in observed data that we can measure this accelerated amyloid production rate and aggregation rate. So here showing a faster accumulation of amyloid in individuals with Down syndrome compared to our Wisconsin Registry for Alzheimer's Prevention cohort. And in a more recent paper that Dr. Zammit and I are working on, we also see this when we apply SILA to understand these overall amyloid accumulation trajectories, where we're seeing Down syndrome on a shorter timeline. When we apply these methods and contrast Down syndrome to our local RAP cohort, again, we can clearly see this difference in amyloid positive age. So amongst those that became amyloid positive in these studies, the Down syndrome group became amyloid positive on average just over age 40, whereas in this subpopulation of RAP, we're seeing people becoming amyloid positive in kind of their mid 60s, late 70s or early 70s. And this stark difference is depicted on the right here in these Kaplan-Meier plots, again, showing kind of these median amyloid-free survival curves with a really large separation of almost 3 decades. Perhaps more interestingly now is contrasting plasma phosphatau-217 and tau PET outcomes across these groups. So the like pink or salmon color being the Down syndrome group, and the teal color being our neurotypical RAP population. So these are showing biomarker positivity along the age spectrum. So again, we see this accelerated pattern in plasma and in tau PET outcomes with respect to age. But more surprisingly is if we put these on an amyloid-based timeline, we see that a suggestion, although this was not significant, that individuals with Down syndrome after amyloid positivity develop biomarker positivity faster. So here with plasma phospho-tau217, and then these were significant in the mesial temporal and universal ROIs for tau PET positivity. So again, in individuals with Down syndrome, we're not just seeing kind of this earlier shift in when people develop positive biomarkers, but even after amyloid positivity, we're seeing a shorter time frame from amyloid positivity to when these other biomarkers become positive. And another thing that stood out to us was in Down syndrome, we don't really see that staggered effect of amyloid positivity, phospho-tau positivity, and then tau PET. We're really seeing these events kind of happen almost coincidentally, I would say within the margin of error here. So the other thing we were fortunate this week, we finally got an NOA for this. So Matt and I will continue to study this, but in gearing up for Down syndrome clinical trials with anti-amyloid therapies, it's really looking like we're going to have to treat very early on in order to be able to affect this disease in Down syndrome. So this is one of the things we're trying to understand. And then on the right here, again, just kind of showing these in a different way that in Down syndrome, we really see the plasma and mesial temporal and neocortical tau all kind of start to become positive really along the same timeline, whereas in RAP, we see this kind of staggered progression pattern. And the other thing that's fascinating is a paper also that was published from using the same or similar data from this Alzheimer's Down syndrome consortium. We see that kind of this uptick in plasma phospho-tau and mesial temporal and universal tau PET also tends to correspond with the inflection point for when we start to see cognitive decline in individuals with Down syndrome. So again, thinking of dementia risk models, it really seems like in Down syndrome, we have a pretty limited window to intervene. So to summarize all of my findings here, several biomarkers, Alzheimer's biomarkers, are highly predictable once we get above abnormal levels. We've shown this with amyloid PET, tau PET, with some caveats about clinical progression and region selection, as well as plasma phospho-tau217. We do need to kind of continue to evaluate the extent to which we can apply a model in other populations, as observed with the Down syndrome study. The other thing that's very nice about being able to model these things with high accuracy is that we can now estimate when biomarkers became abnormal at an individual level. And we're really hoping that this will drive discovery of factors that accelerate disease, as well as allow us to generate dementia risk models that are easy to intuit and are based in years of exposure. And then the other kind of consistent thing that we're seeing across all of these studies is that people that are older when they become biomarker positive have a shorter preclinical time frame. So when we're thinking of lifetime dementia risk, we really need to kind of weigh competing risk of death of other causes with kind of the shorter timeline that we observe for individuals that are older with positive biomarkers. In terms of kind of some future and ongoing work, the big thing we're working on right now is a large multi-cohort study with over 5,000 participants from multiple cohorts to develop a standard amyloid and then hopefully tau-based biomarker clocks that will have a dementia prognosis component. Continue to characterize consistent sources across multiple cohorts and also understand the extent to which these models will generalize across different population groups. So we have an ongoing collaboration, for example, with the Health and Aging Brain Study, Health Disparities at University of North Texas, looking at this across ethno-racial groups. And then lastly, really pathological correlates are going to be the key for us to really understanding these longitudinal biomarker measures and putting them into context. So with that, I will thank you all for attending and for your kind attention. And again, I want to thank everybody that's contributed to these studies. It's a huge privilege to be able to present all of this work, and we have such amazing teams here at Wisconsin and outside of Wisconsin that make it possible.
Great. Thank you, Toby. Maybe you can stop sharing for a few minutes. We take some questions. And I do remember your start. If I remember talking to you in your basement with the drum set in the background there.
Dark days, positive but dark. Yeah.
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 Yeah, from your and your picture with your grandparents, I think it was more band-like or
The long hair definitely was a remnant of being in metal bands. Yes.
yes, yes, great. So there's a couple of questions. I'm going to see if I could interpret them. Very creative use of data from going from longitudinal to cross-sectional in the past to subsequent clinical changes. So I think what's noted is there seems to be some baseline amyloid and tau measurements being able to measure. Is it ever normal? Is some accumulation part of normal aging, or is it always pathologic?
Yeah, I mean, I think this is something that's changed. Like interpretations have evolved over time, right? I think earlier, maybe 5 or 10 years ago, kind of there was this idea that people can have amyloid but remain cognitively unimpaired, and potentially this is some age-related thing and not a disease. I think largely now that's not really. I think that idea is kind of falling out of favor in terms of these things really do, the biomarkers really do represent pathological entities. And it really seems to be the case that if you have these things long enough, you're going to develop dementia. So, it seems to be kind of like a beating the clock kind of thing, right? Are you going to make it long enough in life that you would develop these things, or are you going to die from some other cause before they can kind of have their effect?
Yeah. So they're pathologic. They're not part of normal aging.
That would be my
That's
opinion,
the
yes.
current
Yeah,
current.
especially the tau measures. There's stuff with like primary age-related tauopathy, but largely, like especially tau PET doesn't really detect that kind of small level of tau.
Great. And then some of your levels of amyloid had some plateauing or perhaps some decline in their levels. Does that correlate with any changes in their clinical phenotype?
Hmm. This part is kind of getting fascinating. It's just kind of the dogma of the field is that amyloid plateaus, right? When we look at observational data, we don't actually see a big influence of this, but it's kind of it's kind of been hypothesized for a while that we can't necessarily catch that plateau phase all that well because people are progressing to dementia and we can't continue to follow them longitudinally, right? So it's, the error bars become pretty large out in that range, and it's a little tough to say kind of what's going on there.
Okay, great. And okay, from Bob Probelski, Bob. FDG PET hypometabolism shows a positive correlation with necrosis, tau deposition, and loss of synaptic density. So have you looked at how FDG PET findings support your findings?
Yeah. We haven't done as much in terms of modeling with FDG, and obviously, yeah, this is highly clinically relevant, right? It's one of the most used diagnostic tools in our tool belt. I would say kind of the consensus in the field, right, is that changes in FDG are probably either co-occurring with tau or a little bit downstream of that. So, I think they are pretty highly correlated in that sense. If we're thinking of kind of prognosis in the preclinical time frame, like David Jones has a lot of work on this to kind of suggest at what point those patterns become meaningful. But I think the other thing for FDG PET is not just with Alzheimer's, right? There's a lot of other dementias that we don't have good biomarkers for, that we can pick up on some hypometabolic patterns in other neurodegenerative diseases. And so there's a really nice, I think, differential diagnosis if you incorporate a few of these tools.
Great. And then a couple of questions, which is, I think, the burning question. So once you've detected amyloid or tau, what are the modifiers of deposition? Lifestyle, hospitalizations or ICU
Yeah.
hospitalizations, et cetera. Do any of these slow it down or accelerate?
So we haven't directly, like we meaning like our local team haven't directly looked at this with these models. There was a trial that was presented this past summer, kind of the US Pointers study, where they did see kind of a modest effect of lifestyle in terms of kind of say delaying progression of cognitive decline. And I think the other thing to keep in mind there is like past work in like Sterling's group, we've seen, for example, like things like Libra and Heart Health and other things that are kind of these big risk models don't seem to really influence the like amyloid and tau biomarkers all that much. But that certainly doesn't mean that they can't influence like the time from amyloid to dementia or somebody's like cognitive health. Like obviously these things are very important for people's cognitive health. So whether or not you have Alzheimer's disease, you should do these things, just to maintain a healthy brain
Maintain
and
a healthy
healthy
lifestyle.
life.
Don't
Yeah.
get admitted to the hospital and don't come into the ICU. I think
Yeah.
those are all generally good guidelines to live
Yeah.
by.
Yeah, I mean, I think it'll be nice. We'll have to look actually for the hospitalization and ICU data. I don't know that there's going to be a lot of that out there, but I think it'll be in terms of people that have like big longitudinal biomarker cohorts. But that's something fascinating to look at.
Yeah, and there's a whole body of literature of what the impact of delirium if you're in
Right.
the ICU and poor outcomes. So be interesting. Um, and then how early do you think we're going to need to intervene? The big question.
Yeah.
Yeah.
I'm I'll say optimistic that I think some of the secondary prevention studies that are going on now, I mean, the more we understand about these longitudinal biomarker progressions, I don't know, I think there's a lot of parallels with cancer where it's like, the earlier you can detect an abnormality, the earlier you can treat it, probably the better off you are in being able to slow or stop that disease. So we haven't been able to stop the disease process yet, right? And I would say I'm hopeful that treating in that preclinical phase, especially before, a lot of tau deposition occurs, is really going to be the key um, to really moving the needle in terms of when people progress.
Well, and that brings us to the next question, which is in patients who have people who have Down syndrome, where they're going to be developing, should you be treating them, I don't know, at birth, at adolescence, like
Yeah, I mean, I think this
early?
is where the blood biomarkers will really be influential. If we're thinking about screening, for example, right? I could envision a world where we might, if people have Down syndrome, we might want to start doing blood tests people in their 30s even, right? And we would typically never do this in the context of sporadic AD. But I think that's the other piece where those biomarkers are coming along. That being said, I don't know, the extent to which we'll be able to move the needle in Down syndrome after we're detecting things in plasma is, we won't know until we're doing trials. So um, but yeah, there's been a lot of conversation about this in the autosomal dominant AD space too, and like the Diane study, right? Looking at people with autosomal dominant mutations and treating. Unfortunately, the drug that they used wasn't as effective at removing amyloid, and so it was a negative. They didn't meet their primary endpoints, but hopefully, with some of these more effective amyloid clearing drugs, that'll maybe shift when we can treat with those.
Great. Okay. All right. Well, I think we are just at time and done with the questions. So thank you, Toby. Thank you, Steve, for the intro. Great work, Toby. An impressive body that you've been able to complete even in challenging times. So many kudos to you.
Yeah, no, thank you, Lynn. I appreciate it.
All right, take care, everybody.